you hear me?
I can turn it off. Hey, thanks everybody for turning in your little statement about yourself. It's always interesting to read people's career goals, and I see shifts in that. There always is a very large group who wants to go into veterinary medicine or regular medicine or some kind of a related career field. In relation to that, I have to say, this class is of course very interesting if you have an interest in medicine or something similar. However, this is a general biology class, and I like to teach the class as general for general biology majors. Every now and then I will emphasize something of medical interest, sure. That's one thing. It used to be a couple of years ago that I always saw a lot of people with an interest in sort of security defense type of field. I don't see that much anymore. The trend is more towards education. People want to be teachers now. Honestly, I think the latter is, that you want to be teachers, I think is very good. There's nothing wrong with working with defense. And what struck me is I have read almost every statement, except maybe I need to do still 20. I saw only one Go Broncos on there. I was surprised. All right. So you should have all had introductory biology. You should have probably read the book by Campbell for Life 102 or a similar class. And the unity of life is something that's striking. This is one of the main points of introductory biology. We like to emphasize that life both is characterized by this diversity, but at the same time uniformity. Uniformity and diversity are both well explained within the concept of evolution. Now, at the cellular level, the similarities between life forms are really very, very striking. And there's just a couple of things to point out. What do all cells have in common? They always have a cell membrane that surrounds them. Okay, so the very simple structure of what the cell is, it's something enclosed by a membrane, right? That plasma membrane, or the cell membrane, is something that all cells share. And all cells share the same mechanism for reproduction and for containing and replicating genetic material. So they all do it based on DNA. I can point with my mouse. Everything is based on DNA. It's where the genes are. And DNA encodes for proteins using an RNA intermediate. The process by which the information in DNA is copied into RNA, we call that transcription. The process by which the information in RNA is used to form proteins, we call that translation. Those processes are virtually identical if you compare different cells. And yeah, there are differences if we look at the eubacteria and the archaebacteria and most eukaryotes. But they're minor. The similarities are much more important. And for instance, one thing, all organisms use these same 20 amino acids to form their proteins on a ribosome. There are some exceptional amino acids that fall outside of these 20, but they're not the most important. So if we look at the chemistry and the metabolic pathways that cells have, metabolic pathways, that means biochemical pathways, the way in which organic molecules are modified into other organic molecules. Those biochemical pathways, they're shared a lot between different organisms. Virtually all organisms have the process of glycolysis. That is the process in which in 10 enzymatic reactions, glucose is broken down to form pyruvate, giving us some energy in the form of ATP, and a lot of intermediates that are used in other pathways. Virtually all organisms have the follow-up process called the Krebs cycle, also known as the TCA cycle, also known as the citric acid cycle, which occurs in eukaryotes in the mitochondria, and it serves a role also in energy metabolism and also the intermediates of the Krebs cycle are very important molecules to make other structures. And then all organisms share essentially the same mechanism of ATP synthesis. I should even omit the word essentially. All organisms have the same mechanism of ATP synthesis. And that molecule, ATP, is a carrier of metabolic energy in the cell, and it's important in all cells. So it has this very universal and central role. ATP is the molecule that serves to temporarily store energy that is released from breakdown reactions in order to fuel build-up reactions and in order to make work possible. Okay, doke. Yeah? So you should know this. You should have learned this in introductory biology, but I'm saying it again, and you should know these things. Okay, let's look at the basic structure of the cell. We'll look a little bit, very superficially, at the other structures of cells. We'll have a more detailed look later. Okay, so here we have a typical, simple prokaryotic cell. What do we have here? Well, this cell is a cell, so it has a plasma membrane. It's surrounded by a plasma membrane, which is indicated here as this sort of two-line structure, maybe 3 lines. I don't know if my reading glasses. I can read it. Yeah. A red line, a blue line, and a red line. That's great. Okay, it has then within that membrane an aqueous compartment that we call the cytosol. And there's DNA, because all organisms have DNA. So there's a simple circular DNA molecule in this bacterium. This is an EM picture of a bacterium like that one. We'll look at it later more in detail. There are other structures in this bacterium, like this bacterial flagellum. We might look at that later, for now it's not so important. So bacteria have a cell membrane, a plasma membrane, that's the same. Cytosol, they have DNA. And they use that DNA to synthesize proteins by means of an RNA intermediate. Prokaryotes, okay. So that's a transmission EM picture here. So having just a single membrane to surround the cell is not the distinguishing property that makes a prokaryote a prokaryote. What makes a prokaryote is the absence of a nucleus. There are in certain bacteria internal membranes. For instance, this guy actually might have 2 membranes. It might have an outer membrane outside of its plasma membrane, if it's a gram-negative bacterium. And there are very important organelles called the cyanobacteria, which make up a significant fraction of the biomass on the planet. And these bacteria have internal membranes called the phylocoids, which they use for photosynthesis. This is just one example. Bacteria can have multiple compartments, but they don't have a nucleus. Okay, so then we have the eukaryotes. The eukaryotes have a true nucleus. So here we have a transmission EM picture again. And you can see a lot of structure in there. I'm going to tell you in the course of next week how this type of picture is made. I always like that section. It's something that I look forward to. So here we see that we have in this eukaryotic cell a lot of structures. But most importantly, we once again have a nucleus. We have DNA, which in this case sits in the nucleus. We have a cytosol, where there's all this other stuff, membrane-bounded organelles that are in there. And we have a plasma membrane. Just like the prokaryotes, there's again a plasma membrane, and there's DNA. This time, most of that DNA sits in the nucleus. And there is a cytosol. So that's, once again, transmission EM picture. Okay, now, in eukaryotes, we have typically a lot of organelles within the cell. And then we can define that interior of the cell as the cytoplasm. So we can say that the cytosol, that's the solution within the cell in which the organelles sit. together with all the organelles, that forms the cytoplasm. So cytoplasm is cytosol plus organelles. Minus the nucleus then, and minus the plasma membrane. I don't know why people come up with that definition. Do I care a ton about this definition? Honestly, no. I don't want to be too much of a nitpicker. Not too interested. Cytosol, cytoplasm is sometimes used interchangeably. But if you really want to get it right, the cytosol is that solution in which the organelles sit. The cytoplasm is just everything within the cell, except for the nucleus. Okay, so I really like this book, I have to say. I've been browsing it a lot, setting up the lectures for the coming weeks. I really love it. I think the pictures are really great. But a picture like this and like that, like here, this picture, artist impression, and this EM picture, it's kind of complicated. And it's important for our understanding that we simplify things a little bit every now and then. As long as we remember that we're simplifying. So, this is a cool shot, I agree, but I like the simpler picture a little bit better. So, if we look at the basic structure of a eukaryotic cell, what can we all see? So, we can see here, we have a plasma membrane. Come on. I want to do that differently. Okay. What kind of cell is this? It's a eukaryotic cell. The what type of eukaryotic cell? It is a plant cell because there's a chloroplast. Okay, so that you remember well. I used this picture in Alice 102 and Life 102. So this is a plant cell. So what we have a lot of organelles here. Importantly, it's a eukaryote, so we have the nucleus, holds the DNA. The nucleus is connected with the ERs. Actually, the nucleus is surrounded by a double membrane system. That's called the nuclear envelope, and we can see it right here, nuclear envelope. And that is contiguous with the ER. So if we are in the ER lumen space here, we can diffuse here into the nuclear envelope. That's a contiguous system. Okay, there's the Golgi apparatus. The Golgi apparatus is derived from the ER, the endoplasmic reticulum. And the ER and the Golgi together serve to produce and modify proteins that later get secreted or that go to the vacuole membrane or to the vacuole or that go to the plasma membrane. So we also have in this plant cell a vacuole. The function of that vacuole is storage, and also it has a structural role in plants because there are salts and other solutes stored in the vacuole. Therefore, by osmosis, it takes up water, and that puts pressure against the cell wall that keeps plants upright, keeps them sturdy. Here we have a mitochondrion involved in energy metabolism. Here we have our friend the chloroplast involved in photosynthesis. We will later see there are many kinds of chloroplast-like organelles that we call plastids. They're all related. Now we have this mysterious organelle called the peroxisome. They're also called glyoxysomes, sometimes in plants or microbodies. Now peroxisomes are characterized by the presence of a specific enzyme called catalase, which is an enzyme that turns over, breaks down hydrogen peroxide. And when we speak about the peroxisome, we can make an interesting point. And that is, why do eukaryotic cells have membrane-enclosed organelles? And the answer is that having membranes creates separate compartments, and in the separate compartments, incompatible reactions can be kept separate. So, for instance, in the peroxisome, there are breakdown reactions that take place. That's the function of the peroxisome. And those breakdown reactions are characterized, they're oxidative, they're characterized by the production of hydrogen peroxide. And hydrogen peroxide is actually toxic when it accumulates in the cytosol. So we want a specialized compartment where that hydrogen peroxide is formed, so it doesn't damage other structures in the cell, and then in that same compartment the enzyme is present which takes care of the removal of this hydrogen peroxide. Okay, so our peroxisomes are given this schematic structure here with this sort of crystalline structure inside, and that is something that you can actually see if you look at peroxisomes under an EM, and it's due to the presence of catalase, which has actually iron as a cofactor in there. Okay, so this was a plant cell. That means we have a chloroplast, we have a large vacuole, and we also have, so we have our chloroplast, we have a large vacuole, and the cell is surrounded by a cell wall, a rigid cell wall composed largely of sugars. So that cell wall is characteristic to the plant cell. Other organisms, such as fungi, have cell walls also. They are based on a different structure. Often it's chitin that plays an important role. It's another type of polymer. Okay, to connect the vacuole and the plasma membrane with the Golgi, we can have vesicles, and vesicles also connect the Golgi apparatus with the ER. We will look at that system later in the class. It's together called the secretory pathway, or the endomembrane system. Okay, so that's a plant cell. Now, I call this a key diagram. And what that means is that I think it is fair game for me to ask you a question about this diagram on an exam. You should be able to fill in the names of these organelles if I give you that diagram. It's unlikely that I will ask you to draw structures like this, and that is really because A, myself, my eyes are not that great, and B, I don't want my TA to grade for 200 different drawings. And I am aware that the chairs in this room are extremely comfortable, but the desks are very small. So I think excessive drawing is not what I would expect. At some instances, I will ask you to do some drawings. You will know. Tell me. Okay, another key diagram. Here we have an animal cell. Let's say a typical mammalian cell it could be. So we find a lot of the same structures. We find the nucleus. It holds most of the DNA. You see there's the endoplasmic reticulum. It's contiguous with the nuclear envelope. There is Golgi apparatus, named after, I always forget his first name. I think it was Camille Golgi, I think it was. Golgi was one of those underappreciated scientists because he lived in northern Italy at the end of, I think it was the 18th century, early 1900s, thereabouts. What matters is it was way before there was electron microscopy. But this guy has very good eyes, and not only that, he has very good skill. He worked out all kinds of schemes to stain parts of cells. He was the first to see the Golgi apparatus in a light microscope, which is an amazing feat, believe me. Okay? So that's why it's named after Mr. Golgi, therefore capitalized. Right. Here we have a lysosome that's analogous